Everybody is doing well. So this is the Eye Physiology lecture for Year 2 students. So these are the learning outcomes that I picked out from your lecture. This is in chronological order according to the slides of your lecture, but personally I did not go in chronological order in my explanation because, for example, she directly goes into clinics and I don't really like that method, so I did clinics the last part, and it's just not in chronological order, okay? Before we start, I just want to explain this region right here. So you don't have to look at anything here. This is just for me so that I can recognize and my own ideas so that I don't confuse myself. And when I write the numbers, these numbers correspond to the learning outcomes that I have just mentioned a minute ago. Now, first things first, to start with the retina, we have to know the layers of the retina, right? So backward to forward, okay? The first one is the retinal epithelium. It contains melanin, and it's connected to a vascular wall right here. You can see. Oh, it's right here. Yeah. So this vascular wall, through the retinal epithelium, provides nutrition for the products right here, right? So melanin in the retinal epithelium is responsible for absorbing excess light. So as you see right here, we have light coming in this direction, and these cones and rods are supposed to absorb the light. Now if there's excess light, it's absorbed by the melanin, right? Now the second layer is the photoreceptor segment, pros and cons. Then we have the third layer, outer nuclear layer. It's composed of the nuclei of the photoreceptors. Then we have the outer plexiform layer. Plexiform comes from the Latin word braid, and it looks exactly like a braid. If you want to see the terminals, how they're connected. And then you have the inner nuclear layer, which contains the nuclei of the bipolar cells, horizontal cells, and amacrine cells. Then you have the inner plexiform layer, of course, another braid between the bipolar cells and the ganglion cells. Then you have the ganglion cell layer. This is basically the ganglia or the nuclei that are gonna form the optic nerve. These are the nerve fibers, and the last layer. Okay? So we said the role of melanin. Now what's the role of horizontal and amacrine cells? Right here, horizontal and amacrine cells are related to something called lateral inhibition that we're gonna explain later on. Direction of light and electric impulse. So basically, if you look right here, light, as I said before, goes from here to there, right? And then it's absorbed by these photoreceptors, the rods and the cones. And after that, they generate an impulse, right? This impulse goes in this direction, okay? You should understand that. Now, what is EPSP and IPSP? This is a very important point because action potential in this region is not formed unless it's in the ganglion cells. These, we have either EPSP or IPSP. Now, what does each mean? EPSP means excitatory postsynaptic potential. It means that these are excited. Okay, they're conducting an impulse, but it's not an action potential. IPSP, on the other hand, it's an inhibitory postsynaptic potential. It means that they're not excited, they don't want an action potential, okay? But only true action potentials are here. Only. There's no action potential up here, okay? You have to know this by heart. Now, moving on. So basically, if we zoom in on the retina on this side, there's a kind of a depression called the fovea. Fovea is responsible for something called the visual acuity. Now, what does acuity mean? It's basically sharpness. So, for example, let's say you're looking at a water bottle and then you want to read the nutritional facts. So, through the fovea, you can actually comprehend and read and have visual sharpness for you to comprehend the nutritional facts, right? So, basically, does it have the same number of layers as the retina? Of course not. As you can see right here, these are like 6 or 8, and this is just 3, I think. And it's only cones. It has a lot of cones in this region, which makes sense because we want sharpness, right? Now moving on, so this is a rod, okay? We have to label this rod, okay? So we have the outer segment, the inner segment. Now, in the outer segment, we have the pigments. If it's a rod or a cone, we're gonna have rhodopsin in a rod and photopsin in a cone, right? Now, if you're gonna ask me, why do we have a lot of discs right here, right? Well, that's because the more discs you have, the more increased surface area you have, and the more ability for you to have more pigments. If you recall from gastroenterology, in our colon we have, on the cells, we have these villi, right? These villi increase the surface area of absorption. And it's the same concept here. The more discs you have, right, the more surface area you have to add a lot of pigments, right? That's it. As for the inner segment, you have a lot of mitochondria. Now you're going to ask me, why is there a lot of mitochondria here and not here? Well, basically, when this is not stimulated, this part is leaky to Na+, right? So this goes in without any help, or it just goes in, okay? Now, in this inner segment, there's a lot of mitochondria because to maintain homeostasis, the cell kicks out the Na+, right? So we need ATP so that we can activate the pump to kick out Na+, because it's coming in and no one's talking to it. It's just coming in. There's nothing that's there's no security guard here to not let the Na+ in. So it just goes in, right? So we need to kick it out to maintain homeostasis, okay? So what is receptor potential? Receptor potential is how many photons or light, okay, a rod or a cone needs to activate itself, right? So let's say a cone was hit by 1 photon. Do you think that's enough to activate it? Of course not, right? So if you compare the threshold of cones and rods, of course cones will need a lot of photons to be activated, and the rods won't need that a lot because they work in the dark, right? Now, moving on to where is the blind spot, right? There's a blind spot. So this optic disc where the optic nerve forms is actually the blind spot. In this place, there's actually no cones, no rods, whatever. Blind spot. Review the properties of rods and cones together, right? So rods. Sensitivity to light, it's low threshold. What does that mean? It means that it needs low amounts of light so that it can be activated. Even low amounts of light can activate it, right? Now, of course, it's night vision. It's scotopic vision or night vision, right? Now, cones, they have high threshold. It means that they need very bright lights for it to be activated. And of course, it's day vision or photopic vision, okay? Now, moving on for acuity. Rods have low acuity and cones have high acuity. What does that mean? I want you to think of the rods as your ability to see, and cones as your ability to perceive, okay? For example, you can see a water bottle from a distance, but when you come closer or when you use your cones, you can see the color of the tag from the outside and whatsoever, okay? No, now, rods are not present on the fovea, and cones are present on the fovea, of course, as we said before, because fovea is for visual sharpness, right? Now, I'm going to explain these later. So, color vision, of course not, and cones, of course, yes. Now, I want you to look at these. This is the temporal retina, the nasal retina, and we have a central retina for the visual fields, right? So, where is the cones most dense on the retina? Of course, on the center, right? And on the peripheries, these white ones are the rods, okay? And this makes sense because if, let's say, for example, for peripheral vision, you are sitting right now at your desk and your laptop is on your left, okay? Now, with using your peripheral vision, you can perceive that your laptop is right beside you, but you can't actually know what's written, right? Because this is the rods working peripherally, يعني, which means nasal and temporal, right? But then when you move your face towards your laptop, you're looking centrally at your laptop, you can exactly know and point out what you're reading and writing, which is central, right? Moving on, let's take the example of a rod, okay? So, as we said before, we zoom in on the disc because they're concentrated on the discs, and we see this. This is the lipid bilayer. This is the transmembrane protein called opsin, or I think it's called scotopsin. Yes, scotopsin. It's synonymous to opsin. They're the same thing. They're just synonyms, okay? So this is the transmembrane protein, and this is the retina that is found within the opsin, and the whole thing is called rhodopsin, which is the pigment of the rod. Now, for cones, it's called the cone pigment, and it's formed of retinal, of course, as well as photopsin. In this case, it's opsin for rods. In cones, it's photopsin, okay? So photopic vision is for day vision, meaning cones. And scotopic vision means night vision, meaning rods, okay? Now, what is the role of retinal that is found inside of this rhodopsin? So retinal is actually supposed to absorb the light that is coming in. Let's say there's a photon, it absorbs it, and then it changes shape. Or it's called a conformational change, okay? So it changes from 11 cis to trans. And I don't really recommend going into details on how this changes, because I think you already took that in biochemistry and it's a topic of biochemistry, but I only want you to know that it changes to this. Of course, opsin, or this pigment, rhodopsin, is responsible for dark vision or night vision, but photopsin is responsible for day vision. Now you're going to ask me what colors does it have, or what colors does it absorb? Well, it absorbs 3 wavelengths. They are green, blue, and red. Moving on. So as I explained before, when the rod is not activated by photons, okay, to maintain homeostasis, what happens is that there's no guard here, so Na plus is leaky and it goes into the outer segment. And in the inner segment, there's mitochondria that pumps out this, or kicks out this Na plus, okay? This is just non-active, normal, maintaining homeostasis, okay? Now moving on to what happens exactly when we have a photon. Before it's activated, there's something called cyclic GMP that is bound to the sodium channels on the outer segments and keeps them open, okay? It's naturally leaky because of the cyclic GMP that keeps them open. Please keep this in mind, okay? Because it's gonna matter right now. So basically what happens is that a photon hits the retinal, as I said before, and it absorbs it. When it absorbs it, there's a conformational change. Look, it changed. And then T, which is transducin that is bound to retinal, when it changes its conformation, becomes unbound and it's activated. When transducin is activated, it goes and activates phosphodiesterase. Now, when phosphodiesterase is activated, it goes and breaks down cyclic GMP. And as I just mentioned, cyclic GMP, what does it do? It keeps the sodium channels open on the outer segments. Now, what happens? No cyclic GMP, no open sodium channels. So what happens? No sodium coming in means no positive charge, means negative charge, means hyperpolarization, right? As you see right here. So as I have explained before, this means it's IPSP. IPSP meaning inhibitory postsynaptic potential. There's no such thing as action potential in this region. Now, there's multiple resources on what happens with glutamate, right? Now, it would make sense that if there's IPSP, means that there's no conduction of electricity, means there's no glutamate. This is what it says in your lecture. But other sources also say that even though there is IPSP, a little glutamate goes out, a little, not as much as EPSP, okay? And this actually activates the bipolar cell, okay? The little glutamate, it activates the bipolar cell, okay? And this causes an EPSP in the bipolar cell, and it activates the system. Now, in your lecture, it says exactly that there is no glutamate, but what happens is that it's a defense mechanism and the bipolar cell is activated. But for me, it made more sense when another resource said that there is a bit of glutamate that undergoes EPSP and sends it to the bipolar cell. But to each their own opinion, you should follow what you think is best. So moving on to electrotonus. What does exactly this word mean? Electro means electricity, tonus means tension. Doesn't make sense. Anyways, so it is the passive spread of electrical currents along cytoplasm of neurons and their axons without generating action potential, EPSP, as I said before. Allowing graded signal. What does graded mean? Graded means it adds on each other. For example, the more EPSP you have, the more ATP you're gonna have, right? So I'll go back to the figure so that I can reiterate and to make it easier for you. So let's say this has EPSP. The cone has EPSP, and this has EPSP, and this has EPSP. When you add all of these, okay, this all contributes to the action potential that's going to happen here, okay? This is my understanding of graded potential or electrotonus. So now, moving on to adaptation. It's what I skipped on the table, back up there, okay? I'm going to explain it right here. So let's say we have light. So, as I said, just explained, the photopigment is degraded. 11-cis becomes trans-retinal, as I said, right? Now, I moved from light to dark. I'm in the cinema. I moved from the lobby to the showroom, of course. So I'm moving from light to dark, which means that my degraded photopigment is going to have to reform itself or to connect each other again, okay? That's just basically it. It's just like this. And when I move from dark to light, for example, I finished my movie, I'm going back outside to the lobby where there's light, so my one piece red photopigment is gonna be degraded again, okay? Now, if you're gonna ask me which adapts first, pros and cons, I'll give you an example of you in a theater, okay? You are in the lobby and cones are prominent, of course, because there's lights and there's lights everywhere. It's not dark, of course. And you're walking to the cinema room and it's suddenly dark. Of course, your cones adapt immediately so as you can see your seat and not fall and embarrass yourself. So basically, your cones are the ones that were working first. And then it's like you shock them. You shock them and they adapt immediately so that you don't fall, so that you have visual perception. And then you are guided to your seat and you sit down and the rods start adapting slowly. They start adapting slowly. 20 minutes into the movie, 10-20 minutes into the movie, your rods should be fully working, helping you see details, okay? So, of course, cones adapt first, then rods. Moving on. So what are the types of nerve circuits? There's two. There's a slow one, a fast one. A slow one starts like the message, I mean. When I say slow, I mean the message, okay? It goes through cones and rods, then to the horizontal cell, from the horizontal cell to the bipolar cell, from the bipolar cell to the amacrine cell, from the amacrine cell to the ganglion cell. It's a slow one. But a fast one goes directly from the, this is a cone, to the bipolar cell to the ganglion cell. This is a fast one, okay? Now you're going to ask me, what is lateral inhibition? Okay, if you look at this picture, you're going to see a place of light and a place of darkness, right? Due to lateral inhibition, you can actually differentiate between darkness and lightness, right? So that is due to amacrine and horizontal cells, of course. So as you can see right here, let's say this circuit has been activated by photons, which you're seeing right here, right? What happens is that this horizontal cell deactivates this, or I don't want to say deactivates, it inhibits the circuit that is right next to it. That's why you can differentiate between the light here and, for example, the darkness here, right? They're right next to each other. So this one should be activated and this one is deactivated so that you can differentiate between lightness and darkness. Makes sense. As for the visual pathway, to be honest, this is anatomy. You should know this from anatomy. But in physiology, what you should really focus on is what are the important regions for eye and brain connection, right? There are, of course, the superior colliculus, geniculate body, and the visual cortex. So you're going to ask me, what is the difference between a primary and secondary visual cortex? Well, a primary visual cortex helps you recognize what you're seeing. For example, oh, I'm looking at this is a scarf. And then my secondary visual cortex is gonna help me actually recognize what's on the scarf, or the quality of the scarf, or the material of the scarf. For example, it's polyester or it's wool or whatever. You know what I mean? Now, moving on to voluntary and involuntary fixation. So if you look at this picture right here, I was confused at first. So I decided to have voluntary fixation because it confused me. I thought it was Shrek, but then I looked at this. This is Mike Wazowski's body. So I used my voluntary fixation, or my frontal and premotor areas of my brain, to focus and try to decipher this stupid picture that has been photoshopped. And after that, when I realized it was just an idiot that photoshopped this picture, it became involuntary, and my occipital cortex took the handle. Okay? Moving on. Saccadic movements. Saccadic movements is when your eyes jump from something to something. So if you look at this picture, your eyes are gonna jump from this flower to 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 this and to this. So this is exactly what saccadic movement is, okay? Or when you read a book, when your eyes jump from word to word, that's exactly what saccadic movements are as well, okay? Another scenario I can talk to you about is when you're in a moving car, okay? And when you look out your window, you can actually see that your eyes are jumping from figure to figure, right? For example, let's say you're seeing cars, right? So your brain is actually gonna fixate it and show you each car that your eyes are jumping from. But the background won't be actually, it won't be clear. If you just look at this picture, you can see the car and you're gonna see the other car, right? But the background is not fixated. That's the brain. The brain actually fixates the image of this, but it doesn't fixate the image of the background, okay? Moving on. So what is parallax? I would like to say that parallax is just perspective, because it's not true. What I mean by that, if you're in a car and you're looking out of your moving car, the stuff that are close to you, so let's say your car is close to another car, right? It's gonna seem like the other car is going fast, but that is not true. That is just perspective, okay? Now, why in parallax is binocular vision more accurate than mononuclear vision? So let's say you are moving and an object is moving right in front of you, okay? If you close one eye, you're gonna see in a certain position. And then if you close the other eye, you're gonna see in another position. That is the two perspective of the binocular. But in mononuclear, monocular, you have only one perspective. So it's always better to have both perspectives, right? Because actually, when you have two perspectives, this helps your brain better perceive the distance between you and the moving object. Okay? Moving on. So now I think we're very near the end. Now I'm going to explain learning outcome 2 and 8. This is the clinical part of this lesson. So basically, what is a microaneurysm? These cases are related to diabetes, okay? So you have chronic high blood sugar. This damages the endothelial cells. Now, where are the endothelial cells? In vessels. This is a vessel. The cells on the inside are called endothelial cells, okay? So when you have chronic high blood sugar, this will damage these vessels, okay? And that leads to their swelling. If you look at this, there's a swelling. It was like this and then became like this. That's an aneurysm, okay? It's the hallmark sign of diabetic retinopathy, okay? So the other thing is cotton wool spots. You can see it right here. This is a wool spot. This is a wool spot. This is a wool spot. So of course you have diabetes mellitus. This blocks the vessels. These vessels, they become blocked, Okay, so no blood, no nutrition, right? Makes sense. This will lead to what? Death of the nerves. And the white thing is actually dead nerves, which is very sad. Now, moving on to retinal detachment in diabetic eyes. It's basically, I don't think it's related to diabetes, or is it? Let me see. So it's the separation of the retina, as you can see right here. This is the retina. I don't know if I can zoom in. Yeah, this yellow thing is the retina. It actually detaches from the underlying tissue, which is like the choroid, okay, and from the underlying pigment epithelium, right? So the underlying pigment epithelium and the choroid, as I explained in the beginning of this lecture, are the source of nutrition. So, as I said before, diabetic retinopathy does not directly cause retinal detachment. Diabetes can contribute to conditions that increase the risk of detachment, okay? So, sorry, I'll go back to this. Retinal pigment epithelium and choroid, when the retina is detached from them, this is the retina and this is them, they're detached. It will not receive the nutrition it needs. So, what do you think happens to the signal transduction to the optic nerve? Of course, it's going to be, the efficiency of it is going to be very low, right? That's all you have to know. Moving on to color photochemistry. So we have blue cones. They best absorb blue color at this wavelength. Green cones at 535. Red cones at 570. Now, if you look at this figure, and you're like, okay, so we have red, blue, and green cones. What if I want to look at yellow colors? How does that happen? So basically, yellow is right here, which is between actually 500 and 600. Now, as we said before, 500 and 600 is between the green and the red. So basically, yellow is a mixture between the red and, the green and the red, right? That's how you see other colors, okay? Now, the last thing, which is the Ishihara test, the color blindness test. I wish I can actually help you with this test, but personally, which is very funny, I am colorblind, so I can't really help you with the numbers right here. So feel free to check yourself with another person that is not colorblind. And I wish you the best on the exams. This was the last thing that I wanted to explain for Eye Physiology number 2. I wish you understood everything, and if you have any question about anything, please don't hesitate to ask me. I think you can ask me on my number on WhatsApp, and if you don't have that, please email us and I'll send you my number.